Hello everyone and welcome to this class. This is the continuation of the apoptotic pathway, so it's the second class. Okay, so just follow this hand pinning right here, and I'm also going to write some words in between to explain you better. Okay, so we were talking about the cell death mechanism, right? So let us start with it. The cell death mechanism. Okay, I was talking about apoptosis, necrosis, and autophagy. Okay, necrosis was accidental or shock cell death which happens due to accident, and autophagy where when the body cells eats up their own cells is called autophagy, while the apoptosis was programmed cell death. Okay, it was programmed, it was planned, that's why it is apoptosis. So we will continue with what we are going to continue in this topic, that is first, which is intrinsic pathway. This apoptosis is classified into two parts, that is intrinsic, and second is extrinsic pathway. Okay, so the intrinsic pathway, as I have written here, it depends on mitochondria, while the extrinsic pathway, it depends on cell surface death receptors. These are some receptors found on the surface of the cell. That's why it is, you can say, extrinsic pathway. Here is the chart showing both the pathways. Okay, so as you can see in this chart, there are two ways in which a cell can enter apoptosis. First is intrinsic, this one, first here, intrinsic pathway, and second one is extrinsic. Okay, so we are going to talk about intrinsic pathway in this video. So this intrinsic pathway can occur due to stress, virus infection, and some development defects also. And as it is shown in the figure, here is some words. This is not important. We will directly go to BCL2, then we have cytochrome C, which is important, and here it is mitochondria. Okay, since the intrinsic pathway is dependent on the mitochondria, that's why mitochondria is very important in it. Okay, we will skip these parts first, and I'm directly going to explain this from the cell. Okay, consider this to be a cell, this right here cell, this is plasma membrane here, and inside the cell there is nucleus. Now nucleus has some DNA. Let us say this DNA somehow got damaged, can be from UV or some other factors of DNA damage from chemicals, etc. Okay, now due to the damage of DNA, p53 protein is activated, and this p53 protein, what it does is it recruits BCL2 family and also affect the backs also. Apart from this, P53 here, it will halt, stop the cell from entering the next cell division stage so that wrong information will not transcribe and translate. Okay, so that's it, right? Now we will move to what I have written earlier, which is the starting of this intrinsic pathway. Okay, so in the intrinsic pathway, also called mitochondrial apoptotic pathway, okay, it starts from inside the cell. See, this is a cell here, and this cell contains mitochondria. And what happens is when a cell suffers from a stress like DNA damage or virus infection or development defect inside the cell, okay, from inside, then some proteins are released from mitochondria. Okay, like as I have told you, like P53, what it will do is it will affect BCL2. Okay, and BCL2 will further activate BAX. This BAX will cause pore formation in mitochondria, right? Then these caspases are activated, which are preserved proteins. Then what happens is these caspases will go and help in apoptosis. Apoptosis. Okay. So the key protein in the intrinsic pathway is the cytochrome C protein. Okay. This is mitochondria, and inside the mitochondria they have cytochrome C, which are released out of the cell. These cytochrome C will go and bind to APAF1 protein. Okay. I have drawn this APAF1 protein just like the antibody, but don't consider it antibody. It's just APAF1. APAF1. Okay. The full form of APAF1, APAF1 is apoptotic protease activating factor 1. And what happens is APAF1 is converted into wheel-like apoptosome, right? This right here is a heptamer, and what we call it is apoptosome. Apoptosome. Okay. So this apoptosome will further go and recruit caspase 9. caspase 9. See here, caspase 9. Actually, what happens is it converts pro-caspase into caspase 9. That's how it recruits caspase 9s. And then it will go and further, this caspase 9 will go further downstream and activate executioner caspases, which is caspases 3 and so on. Okay, so as I was talking about BCL2 family. Okay, so BCL2 is a highly reserved family, and I have written sorry, just like caspases, and for the caspases I use the term reserved, but I should have said it is a highly reserved family. Okay, and these are some functions of BCL2 family, which are in mammals they control the release of cytochrome C and mitochondrial proteins, and in other species they sometimes they promote the apoptosis, sometimes they demote the apoptosis, and they even prohibit or blocks the apoptosis. It depends from species to species. Okay, and this diagram I have already explained this to you. Okay, and this is it. So yeah, okay, now you can read these lines yourself, and I'm going to explain it in the end as well, right? See, I have written everything here. That's it. So this is not thank you. Just wait for a while. I need some more space. Okay. Let us say this is a cell. Okay. And here is the damaged DNA with a nucleus inside it. This damaged DNA, what it will do? There comes P53, and it will give the signal such that the cell will not enter. First, not enter into further cycle. Okay, and second, it will give signal to BCL2, and BCL2, what it will do, it will activate the Bax protein, and they will create pore in mitochondria. And due to this pore formation, what will happen is the what was that cytochrome C will release out from mitochondria, and this cytochrome C will go and attach to APAF1. APAF1. Many APAF, not many, actually 7 APAF1, APAF1 plus cytochrome C will form heptamer, and this heptamer will activate caspases. First, it will activate caspase 9, then it will go and activate caspase 3. That's how this caspase 3 will go and do apoptosis. Okay, that's how this whole process works, and that is what I was going to explain you in this video. And thank you very much for this, and we will see you soon.